In this section, we will focus on bacteria. We have more bacteria on and in our bodies than we even have our own human cells. Most bacteria are good, and we have a symbiotic relationship with bacteria. Bacteria are single-celled organisms, and as you know, they are prokaryotic cells, meaning they have no nucleus and no organelles. Bacteria have a cell wall, a cell membrane, ribosomes for protein synthesis, and they have a circular piece of DNA. We call it the chromosome, and when it's coiled up, it's called the nucleoid region. Bacteria replicate by binary fission. This process is asexual reproduction, which allows for no variation in the daughter cells. So the daughter cells are clones of the original cell. One structure we find in most bacteria are called plasmids. Plasmids are really small circular pieces of DNA. They are much smaller than the bacterial chromosome. They may carry genes that are beneficial for survival of that bacteria in different environments. Antibiotics are chemicals that destroy bacteria by targeting the cell wall. Our cells are not impacted by antibiotics because our cells don't have a cell wall. So antibiotics are great medications that we use to fight bacterial infections. The problem is we often overuse antibiotics and the bacteria start becoming resistant to the medications, the antibiotics, and the antibiotics start to not work against these bacterial infections anymore. Using antibiotics doesn't change the bacteria. Rather, it selects for the bacteria that are already resistant to a certain antibiotic. Then these bacteria are the ones that survive and reproduce. So over time, all of the bacteria become resistant to that antibiotic, and now our antibiotics won't work against certain bacteria. We said that bacteria replicate asexually through binary fission, which provides no variation. The ultimate source of variation in all organisms is really mutation, which can be good, bad, or neutral. So we're going to talk about 3 ways that bacteria can obtain some genetic variation. The first of the 3 is called transformation. This is a very random process. What happens is when a bacteria dies, it may lyse or burst in the environment and just release their genetic material and plasmids into the environment. So randomly, a plasmid or a piece of genetic material might end up inside a bacterial cell, and that plasmid might have genes that are beneficial to the bacteria. The second way bacteria can obtain genetic variation is called transduction. This is also a completely random process. Hopefully you remember the viral life cycle and how viral particles are made inside a bacteria cell, and they put the viral nucleic acid inside the viruses. Well, sometimes a mistake is made, and what happens is bacterial DNA ends up inside the viral particle. Then that virus goes and infects another cell, but it's actually injecting bacterial DNA, not viral DNA. So that cell is not being infected at all. In fact, if the genes or any information on this DNA is beneficial, it is actually advantageous to the new bacteria. And you can see it will incorporate it into its genome. The third way bacteria can obtain genetic variation is through a process called conjugation. Conjugation is a less random process. It's very directed at variation. Some bacteria have these things kind of sticking off of them called papilli. The bacteria that has a pilus is called a donor bacteria. And that pilus actually connects two bacteria together and forms what we call this mating bridge, where their cytoplasm actually connects. And that donor bacteria can transfer plasmids or parts of their genetic material to the recipient bacteria. So you could imagine if, say, this plasmid here had an antibiotic resistant gene on it, you could see how bacteria could easily transfer antibiotic resistant genes from one cell to another. All of the mechanisms we talked about are ways bacteria can obtain genetic variation. So this slide shows what happens when that new genetic material gets into the cell. It literally recombines and becomes part of the chromosome. If it's a plasmid that was added to the cell, then the plasmid stays as a separate entity. But that bacterial cell will express any genes that have come into the cell, and you can see how easily it would be to make many more bacteria resistant to the antibiotics that we commonly use. I hope that was helpful.